So our first speaker is Chris MOXHAM. So he's CEO of Rarebase, which is a biotech company, really is doing platform for drug discovery. Right. So Chris.
Well, while we're waiting, I'll say thank you very much to Mike and the SRF team for the invitation. Certainly very pleased to be here today telling you about the work that we've been doing in partnership with SRF at Rarebase. So most people are familiar with the statistics on this slide, and these are statistics around really the entirety of rare disease. But as a drug hunter who's been doing this almost 30 years, to me, this is a hyper-rich arena for drug discovery, and it deserves a lot more attention. And I say that because the human genetics have told us 3,800 times what the root cause is of these rare diseases. A single human genetic linkage between this gene and that disease. And I think we've all seen in oncology that when we understand that, there's a flood of research and drug discovery that occurs to find treatments. And so to me, this is a hyper-rich arena for drug discovery, and that's why Rarebase has been formed today. The way in which we go about that and to try to find small molecules that rebalance root cause gene expression is that we use a transcriptomics platform, and we do that for good reason. I've already told you about this 100% certain gene-disease linkage, but we can interrogate virtually all of these genes all at the same time through massive parallelization using RNA sequencing technology. And of course, we all understand and are starting to see that there's a golden path that's being laid down in the context of clinical development. Now, maybe today it's 14 karat gold, and in the future we want it to be 24 karat gold. But I think there's movement in the context of it at the FDA in terms of incentivizing further clinical development in this space. But as a pharmacologist, there's really strong rationale to look at the transcriptome. The first is that this is in fact how many, if not virtually all, drugs work in terms of ultimately mediating their therapeutic effect. That is, they modulate gene expression. The other is that there's high translatability of looking at transcriptomics at the bench, if you will, in the lab, and translating that into patients. As we know, many human diseases are characterized in terms of how the transcriptome, that is how gene expression is altered. Diagnostic platforms are built on transcriptomics. Biomarkers are built on transcriptomics. And so this is a highly translatable currency, if you will, that we're measuring in the lab that has already shown great ability to translate into patients. So what we've been doing at Rarebase for the last couple of years is two types of screening with the platform. The first is screening a small molecule library. We'll call it a chemogenomic library, and that's a special type of library that includes FDA-approved drugs, it includes molecules that have been in clinical development, it includes molecules that cover the druggable target space very well. And there's a strategic reason to do that, because when you find something, it's much more actionable, right? There are fewer steps to optimize that molecule to get it into the clinic. And you can see in orange, the data that we've been generating over the last 15 months. We generate a lot of data. In a simple screen from 4,000 compounds, we have 160 million data points. I used to run high-throughput screening at Eli Lilly. In a single screen, we used to generate maybe 200,000 data points in a screen against one target. We've now screened 20,000 genes all at the same time in response to each drug. That's because of the depth of sequencing that we're conducting. So we're looking at drug response across nearly the entire transcriptome. And this is a very unique data set that we're generating. And importantly, in this data set, we detect 85% of all the known root cause genes. So 3,000 targets all in one shot, right? No one screens 3,000 targets all in one shot. This gives us great potential for discovery across the entirety of the rare disease landscape. The other thing that we've been doing is screening more privileged molecules, if you will. That is, molecules that have been in clinical development that failed in some other indication due to lack of efficacy, and now we're profiling those in our platform to see if we can find fast path opportunities to get back into the clinic with a molecule that's already had a human experience, right? There's an understanding of safety and tolerability. And so the work to date has really produced great yield. We have hundreds of confirmed actives, if you will, across hundreds of rare disease root cause genes. And we've also identified among these clinical stage assets over 50 different potential opportunities to move back into the clinic. So it's an exciting time, and certainly we're very pleased with the progress that we've made in partnership with SRF, and I'll start to tell you about that now. The other thing we're doing with all of this data is using machine learning. Right? And so the Rarebase drug gene atlas that we've generated allows us to link molecular structure to modulation of gene expression. And so we're today now have multiple machine learning models in place that are starting to predict novel structures that would tune gene expression in the appropriate way to, again, further build upon the data that we've already built in the lab, and now start predicting novel structures that would be the basis of new chemical entities that could be pursued for drug discovery. So the work we've been doing with SRF starts with characterizing actives that we found from our screening activities in patient-derived models. And so there are 4 different models that we've been working on: FM38, FM40, 41, and 45. The first, these patient-derived models, they span different types of mutations. The thing to understand is that 2 of them include truncating variants shown in blue on the far right, and for FM40, FM38 in the bold black type. And then down below in the missense variants, we have FM45 in the plexion homology domain, and in green, FM41, this missense variant between the C2 domain and the gap domain. So with FM38, this was derived, again, a patient-derived line. It was a dual SMAD inhibition that led to the production of the NPCs, and then ultimately these cells were maturated into GABAergic neurons. They express the classical markers that you would expect to see in these neurons, both MAP2, TUJ1, and that are shown here in the imaging. The other cell lines were generated through more of a classical NGN2 induction. These cells are transduced with a lentiviral construct that encodes for the expression of NGN2. NGN2 is induced, and then over a period of several weeks, you can generate glutamatergic neurons. And again, these neurons, through imaging that you see on the far right, express the classical markers that one would expect to see in these types of neurons. And just more imaging characterization here again for the 40, 41, and 45. They express, and here just a larger image, you can see they clearly express TUJ1 and MAP2. And so we are able to generate high quality neurons with each of these patient-derived models. So now I'll tell you a little bit about the screening activities and the data to date. So the protocol is generally shown here. We differentiate the iPSCs into neurons. This is a process that takes about 3 weeks. We then treat in these microplates 1 compound per well. The compounds that we selected were the actives from our screening activities. We test those in 10 concentrations, so that's called a concentration response curve. There's a rationale to do that. And then each of these experiments are conducted in quadruplicate, so we have 4 replicates per treatment. After 3 days of treatment with the compound, we then make a series of measurements. The first has to do with an imaging-based analysis that looks at essentially cell viability and just the phenotypic assessment of the neurons. Have we changed that phenotype in some significant manner? The other is where we deploy our next-generation sequencing platform. So every compound that we test, we're looking, yes, at Syngap, we're also looking at 20,000 other genes, right? So every compound basically goes through 20,000 assays every time we turn the crank in our lab. And after that data analysis and sequencing, we then look at the data analysis in the context of that concentration response curve. And so what you'll see as I start to show you the data, on the x-axis we have the increasing concentrations that we utilize, and then what you'll see is the measurement of fold change relative to the DMSO control treated wells in terms of the data analysis. And there's a rationale to look at concentration response curve data because that begins to help inform dose, right? And particularly when we're talking about FDA-approved drugs, the question becomes, based upon the approved dose, can you achieve the exposure that you believe you need to achieve with that approved dose? And that would be the fastest path to a repurposing avenue. The data analysis workflow, just for those of you that are interested, right, we're translating RNA to RNA counts. What's the relative abundance of any one of the RNAs that we measure? We then, of course, relate that and turn that into a fold change relative to the DMSO control wells. And then as we do these concentration response curves, we're translating that into a concentration response analysis. So this is the data from the actives that we had identified from our screen in the wild type cells that have now been tested in this FM38 patient line. And we have 4 actives that have shown a concentration-dependent increase to different degrees. And so just to orient you to the data in the table, we have the name of the compound, so nortriptyline, rilpivirine, selumetinib, and capecitabine. You have the potency value that was calculated from the curve. You have the delta in terms of fold change, either on the log2 scale or on a linear scale next to that. We have the goodness of fit, the R-squared value. We also then include, and this is exactly what we shared back with the SRF team a few weeks ago, we include the route of administration. Is the molecule predicted to be blood-brain barrier penetrant? Is it on a pediatric formulary? What's the known target of that molecule? And of course, there's always a debate about whether one actually knows all of the targets that a molecule interacts with, but these are the targets that the literature would indicate that they interact with, and what is the approved indication for that molecule. Just importantly, just for the sake of what we do at Rarebase, right? These are research results, right? We are not physicians trying to instruct anybody about what they should do, but really this is a research collaboration. And so you can see, for example, down on the lower left with a molecule like nortriptyline, there is a concentration-dependent increase. The potency of that molecule is around 6 micromolar, at least in this particular assay. There can be variation in that EC50 value, but again, you start to get a feel for the 4 actives that we have identified here in the FM38 patient cell line that all lead to a significant increase in Syngap1 mRNA expression. The other thing we do is that we're testing molecules all the time in the lab, right? And as I told you, every molecule goes through 20,000 different assays, and so every time we test a molecule in these neurons, we potentially have another active that might induce Syngap. And so what we did is we went back and queried our database again around some of the new data that we had generated, and we identified 3 additional molecules that also demonstrated a concentration-dependent increase in Syngap1 mRNA expression. And those 3 molecules are shown here. You can see the potency values all around 1 to 2 micromolar, the fold change in terms of increase, the goodness of fit, the route of administration. These are all orally bioavailable molecules predicted to be blood-brain barrier penetrant. And so this is, a starting point, if you will, for the basis of going down the road of potentially repurposing some of these molecules for use in patients with Syngap1 deficiency. So some of the key takeaways from the work that we've done, we've characterized 4 patient-derived cell models. I didn't show you most of the characterization data that we've done looking at mRNA, protein, imaging, looking at Syngap localization to the synapse, for example, in these neurons. The repurposing efforts have identified 7 FDA-approved drugs that are being further characterized across all of those 4 patient-derived models. And again, we'll continue to query our database to see if we have any other additional actives as we move along and generate additional data. I think the recent presentation that we just saw with the expression of Syngap in RPE cells, we are actually screening our library across multiple different cell types now, which will include the RPE1 cells. So as we look at additional cell types, one, it diversifies the data set, but also gives us additional opportunities as these pathways tend to be conserved. And as we begin to understand how some of these genes are expressed in other cell types that you might not expect, I think there'll be sort of an evergreen opportunity here to discover new potential repurposing opportunities. So with that, I'll say thank you, and maybe we got us back on time. And